All right, today we're jumping into something really cool, protein engineering. We're talking about a field where scientists are literally learning to rewrite the code of life itself. They're building custom proteins to tackle some of our world's biggest problems.
So, how do you even start to improve on what nature's already perfected? I want you to think about proteins as these incredible little machines. They're doing all the heavy lifting inside every living thing. But here's the big question. What if we could take those tiny machines and, upgrade them, make them better, faster, stronger? Well, that is the whole point of protein engineering.
So here's our game plan for this explainer. First, we're going to nail down what protein engineering actually is. Then, we'll get into the why. What are the main goals here? We'll look at the tools of the trade, the techniques they use, and this is a big one, especially if you're studying this. We're going to walk through a super important flowchart for mutagenesis. And finally, we'll wrap up with some mind-blowing real-world examples of this stuff in action.
All right, let's dive right in. Section 1, the foundation. When we talk about protein engineering, what are we really talking about? Okay, so here's the core definition. Protein engineering is all about developing useful or valuable proteins. But the key thing to get here, and this is super important, is how they do it. They don't typically mess around with the finished protein. Nope. They go straight to the source code, to the blueprint, the DNA. They alter the cloned DNA to change the final protein's properties.
And this isn't some isolated science. It's really the next generation of recombinant DNA technology. It's this awesome mashup of different fields, like molecular biology and protein chemistry, coming together. It's where we take what we know about DNA and what we know about how proteins actually work and combine them to create something totally new.
Okay, so we've got the what. Now let's talk about the why. Why go through all this effort? What's the end game?
Well, the goals are actually super practical. For example, maybe you need an enzyme that can work in really hot water without falling apart. Or maybe one that can survive in a very acidic environment. You might want to make a protein super specific so it only latches onto one single target molecule and ignores everything else. Or maybe you just need to make it tougher so it doesn't get chewed up and degraded inside a cell. It really all boils down to tailoring a protein to do a very specific job better.
So that brings us to the how. How do scientists pull this off? Well, they've got a toolkit with two main approaches.
So you've got two paths. On one side, you have genetic modifications. Think of this as rewriting the blueprint before the house is built. It's incredibly precise. You can change a single letter in the DNA code at a very specific spot. The main way they do this is with something called site-directed mutagenesis. Then on the other side, you have chemical modifications. This is more like painting the house after it's already built. You're changing the existing protein directly. It's way less specific and usually only messes with the amino acids on the surface. So when you need precision, genetic modification is definitely the way to go.
Okay, so let's zoom in on that genetic technique because it's the most powerful one. We're going to break down how you actually rewrite a gene. And hey, if you're taking a class on this flowchart is absolutely essential to understand. Pay close attention here.
We're going to focus on site-directed mutagenesis. The name really tells you everything you need to know, right? You're directing a mutation to a very specific site. It's like genetic surgery with a scalpel-like precision.
All right, let's walk through the single primer method. Step 1. You isolate your gene of interest, but you need it as a single strand of DNA. Okay, easy enough. Step 2, and this is the really clever part, you synthesize a short little piece of DNA, called a primer. Now this primer is designed to be a near-perfect match to your gene, but with one tiny deliberate mistake. It has a single base mismatch right at the spot where you want the mutation to be. And for step 3, you just mix them together. Now you might think that mismatch would be a problem, but it's so small that the primer can still bind or hybridize right onto that single-stranded DNA.
Okay, so the primer is locked and loaded. What's next? Well, in step 4, an enzyme called DNA polymerase comes along. This is the cell's natural copy machine for DNA. It sees that primer as a starting point and just starts building the rest of the DNA strand, copying the template. But here's the trick. It builds the new strand with your mutation now baked right in. Then, in step 5, another enzyme called DNA ligase acts like a molecular glue, sealing up the new strand and making a complete double-stranded circle of DNA. And for the final step, you take this brand new molecule and pop it into a host cell, something like E. coli. The E. coli doesn't know any better, it just starts replicating it, and boom, you get millions of copies of your original gene and your newly mutated one.
So that's the how. We've gone through the nitty-gritty. Now for the payoff. What can we actually do with this amazing technology?
Yeah, this quote really nails it. We're talking about engineering proteins that are tailor-made for a specific application. This isn't just theory in a lab. It's about solving real-world problems.
Let's start with something you probably have at home right now, laundry detergent. A lot of detergents use an enzyme called subtilisin to get rid of tough stains. Great, right? Well, there's a problem. Bleach completely destroys it. So what did scientists do? Using this exact technique, they swapped out just one amino acid, a methionine, for an alanine. And the result? A new and improved subtilisin that's totally bleach-resistant, a better, more powerful detergent. And in medicine, the impact is, wow, it's huge. Take insulin, for example. The therapeutic insulin used by millions of people has a tendency to clump together, which makes it less effective. So scientists went in, made a few specific changes to its amino acid sequence, and created a version that doesn't clump. It stays as a single molecule, a monomer. And this engineered insulin, it's way more stable and works much better for patients, a massive improvement.
And the list just goes on and on. Scientists have taken the enzyme T4 lysozyme and added special bonds to make it stable at super high temperatures. They've tweaked a drug called TPA, which busts blood clots, so it lasts longer in your body. They've even taken a protein from leeches called hirudin, swapped out a single amino acid, and turned it into a much stronger anti-clotting drug. It's just incredible what these tiny, precise changes can do.
So, if there's one thing to take away from all this, it's this. By being able to precisely alter a protein structure right at the genetic level, we can fundamentally enhance its function. We're not just stuck with what nature gave us anymore. We can now redesign these molecular machines to do exactly what we want them to do. And that really leaves us with a huge question, doesn't it? We have the power to redesign life's most basic building blocks. We can literally rewrite the operating system. So, what challenges should we solve next? Creating enzymes that eat plastic waste? Designing proteins that hunt down and destroy cancer cells? The possibilities are staggering. It really makes you wonder, what's next on the horizon?